Welcome to Biochemistry Unfolded, from atoms to algorithms, an online podcast on the topic of biochemistry and biophysics. Welcome to Biochemistry Unfolded, from atoms to algorithms, episode 13. Today, I'm diving into a mystery that bridges the gap between the physical laws of the universe and the complex history of biological life. When we think of dark energy, we usually think of deep space and the expansion of the universe. But a groundbreaking new paper published in PNAS by Ezekiel Galpern and colleagues entitled Probing the Dark Energy in the Functional Protein Universe suggests there's a different kind of dark energy hiding right inside our own cells, inside of our proteins. To further understand this, we have to understand what a protein actually is. Proteins are polymers of amino acids that are essentially the workhorses of the cell. These enzymes digest our food, fight off viruses, build your muscles, and even constitute the very connective tissue that literally holds everything in your body together. For decades, the holy grail of molecular biology has been the folding problem. That is, a protein starts as a long, floppy chain of amino acids, and it must fold into a specific 3D shape in order to function. Think of it like a piece of biological origami. If it folds correctly, it works. If it doesn't, it's just a useless tangle of amino acids. And so when protein misfolding occurs, that leads to disease, such as neurodegenerative diseases like Alzheimer's disease. For a long time, we thought the main rule of evolution was simple. Keep the protein stable. Make sure it stays folded. But this new research suggests that folding is only part of the story. There's a hidden force, a dark energy, that is just as important. So what is this dark energy? The researchers define dark energy as the gap between two different types of pressure. First, there's the physical pressure to stay folded and to stay stable. We call this the physical folding energy. If a mutation makes a protein wobbly or unstable, the physical energy goes up, and the protein might fail. Second, there is the evolutionary pressure. This is the history of what has actually survived over millions of years. We call this evolutionary free energy. And so by looking at thousands of species, we can see which parts of a protein Mother Nature refuses to change. But there's a twist. Sometimes Mother Nature protects a specific spot in a protein, even though that spot actually makes the protein less stable. From a purely structural perspective, it looks like a mistake. But from an evolutionary perspective, this is vital. That mismatch, that extra pressure that isn't about folding, is what the authors call dark energy. And so if we look at figure 1 in the paper, this acts as a conceptual map to explain the researchers' main discovery. How to tell the difference between a mutation that breaks the protein's shape and one that breaks its function. The figure compares two landscapes represented as funnel-shaped energy diagrams. That's energy landscape theory. And so there's three different scenarios. One is a normal protein, that's the wild type. Two is a variant that affects folding. And three is a variant that affects function. The folding landscape is shown with a green line, which represents the physical stability of the protein. That is, how well it sticks together in a 3D shape. The evolutionary landscape is shown with a purple line. That represents the fitness. How much millions of years of natural selection have allowed or forbidden a specific sequence. So in panel A, that's the wild type or normal protein. This shows a normal functional protein. There's a natural gap between the physical folding energy and the evolutionary energy. This gap is what the authors call dark energy, or E-dark. It represents the extra evolutionary pressure that isn't explained by simple folding. Now in panel B, this shows a variant that affects folding as well. This scenario shows what happens when a mutation makes a protein unstable. That is, it doesn't fold well. Because the protein is physically broken, evolution also hates it. The two landscapes move together, so the dark energy gap stays roughly the same as the normal version. In other words, this mutation is bad simply because the protein fell apart. And in panel C, now this is what we call the smoking gun, if you will. That's a variant that affects function. This is the most important part of the figure. Here, a mutation makes the protein much worse in the eyes of evolution. The purple line spikes up, but the physical folding stability is not affected very much. The green line stays relatively low. So in conclusion, if the protein is still physically stable, but evolution nonetheless forbids it, the protein must have lost its biological function, like its ability to bind to a drug or catalyze a reaction. This creates a large spike in the dark energy. To put that in simple terms, figure 1 shows that the dark energy is a tool for scientists to find the active parts of the protein. By looking for spots where evolution is much stricter than physics requires, they can pinpoint exactly where the protein's business end is located. So how did they find it? This is where the atoms meet the algorithms. The team used a combination of two very different tools. First, they used a physical model called AWSEM, Associative Memory, Water-Mediated Structure and Energy Model. And you can find this at awsem-md.org. This is a computational tool developed by the Papoyan Lab at the University of Maryland and the Wellness Lab at Rice University, and it calculates the physical forces between atoms. It tells us how much a mutation will hurt or help the stability of the protein's shape. This represents the physical rulebook. Second, they used a cutting-edge artificial intelligence called ESM2. This is a protein large language model, or PLLM, that calculates evolutionary scores that represent how compatible a mutation is with the protein's evolutionary history. Physical folding energies, the delta E of folding, were determined either from megascale, experimental delta-delta G data, or predicted computationally using the AWSEM coarse-grained force field. Just like an AI learns the rules of human language by reading books, ESM2 has read millions of protein sequences from across the tree of life. It knows the evolutionary rulebook. It knows which mutations are forbidden by history, even if we don't know why. By comparing these two rulebooks, the research could then map out exactly where the dark energy is located. They even came up with a way to convert the AI's statistical scores into physical energy units using something they call selection temperature. This allowed them to measure biological pressure with the same precision that we use to measure heat or electricity. By comparing the scales of these two landscapes, they derived a folding selection temperature, that's T fold, to normalize the evolutionary data into physical energy units, which we can read as kcal per mole in chemistry terms. So what did they find? First, they discovered that dark energy is not spread out randomly. It's highly localized. It clusters almost perfectly at functional sites, and that's the active parts of the protein. And these actually do the work, kind of like binding to a drug or cutting a chemical bond in catalysis. So about 25% of the positions in the proteins they studied showed significant dark energy. This means that for a quarter of the protein, evolution cares more about the job the protein is doing than how stable the shape is. Figure 3 presents a large-scale analysis using experimental data to show how dark energy is a widespread and quantifiable feature of the protein universe. And this shows that approximately 25.3% of protein site variants are primarily constrained by their biological function, while the remaining 74.7% are primarily constrained by their ability to fold. Using a Gaussian mixture model, the researchers successfully separated mutations that only affect stability, low dark energy, from those that impair function, that is high dark energy. And they established a threshold of 1.03 kcal per mole to identify these functional outliers. Now, when considering specific amino acids, mutations to cysteine and tryptophan consistently show the highest dark energy, meaning that they are often strictly preserved by evolution for functional reasons beyond just holding the protein together. In contrast, mutations to proline often have a massive negative impact on a protein. But this is almost entirely due to physical destabilization or folding rather than a loss of specific function, and that results in a very low dark energy. They also found what they call enzymatic halos. In enzymes, there's a core of dark energy where the chemistry happens, and that's surrounded by a ring of residues that support that function, even if they make the overall structure a bit more strained or frustrated. So figure 4 focuses on enzymes using beta-lactamase as a primary example to show how dark energy perfectly maps to catalytic activity. The residues that are directly involved in chemical reactions, that's in the active site, show extremely high dark energy, and that often exceeds 3 kcal per mole. These sites are strictly forbidden by evolution, even though changing them might actually make the protein more physically stable. High dark energy is not just found at the exact spot where chemistry happens, it forms a halo around the active site. The intensity of dark energy is highest at the center and gradually decays as you move into the outer layers of the protein structure. The researchers also measured that this gradient across many enzymes finds that functional constraints in dark energy drive 87.6% of the variation at active sites, compared to only 38.9% in the outer shells of the protein. And so high dark energy appearing far from known active sites may reveal previously undiscovered allosteric regions or hidden control switches where the protein interacts with other molecules. So why does all this matter? This is a huge deal for the future of science. If we want to design new proteins to fight climate change or to create new medicines, we can't just focus on making them stable. If we only follow the rules of folding, we might miss the very features that make life work. So by quantifying this dark energy, we can now distinguish between a mutation that breaks a protein's shape and a mutation that breaks its ability to function. This could revolutionize drug design, allowing us to target specific high-energy spots that are most crucial for disease-causing protein to do its job. The universe is also full of hidden forces, and it turns out that our own biology is no exception. By looking at the gap between the physics of today and the history of the past, Galpern and his team have actually given us a new lens to see the functional protein universe. So thank you for joining me in this episode of Biochemistry Unfolded, from atoms to algorithms. Tune in next time as I continue to explore the molecular world, from the smallest atoms all the way down to the most complex biomolecules. If you enjoyed the content of this episode, be sure to like this video and hit the subscribe button to subscribe to my channel. And also hit the little bell icon so you can be updated when new content is posted. If you have any questions or comments, feel free to leave them in the commentary below, and we can try and address those comments or questions as you stay fit. So thanks for watching and stay curious. I'll see you again in the next episode. You have been listening to Biochemistry Unfolded, from atoms to algorithms. Thank you for watching. We will see you again in the next video.